Welcome. Good to see you all in here. Did you have a good holiday? Today's webinar we have prepared as the beginning of a series. There are more to come, so please keep following us. Now let's get starting. So, who's in the program today? So we have Lara, we have me, we have Lara, and of course we have the Nanoimager. This is the agenda for today. So after this short introduction, we get a presentation from Lara, and Lara is an FIS working in the United States. She will be talking about the Nanoimager, the specs and the applications. Lara will be next, and she will be demonstrating the Nanoimager from a remote location. I will be back at the end where we do the questions and answers. All the questions, they can go in the public chat channel. So at the end, we will summarize them and answer them. So, may I introduce Lara?
Hi everyone, I'm Ja. I'm one of the field application scientists. And today I would like to talk to you about our flagship product, the Nanoimager. I'm just going to share my screen. Here we go. And so this is our flagship product, the Nanoimager. And today I'll be talking to you about how we can bring super resolution to a benchtop near you. A little bit about our company. ONI's mission is to help accelerate human discovery by enabling everyone to see and understand the microscopic details of life. And we aim to do this by providing accessible technologies that are clinically translatable, and we aim to promote collaborative discoveries together. We have two headquarters, one in San Diego and another one in Oxford. We have over 100 employees and we're rapidly growing, and we've just received our Series B funding. So let's dive into microscopy. Microscopy is a powerful tool, and it's very important for allowing us to see the finer details in life. But the challenge of microscopy is that conventional microscopy is limited in both sensitivity and resolution. So super resolution can help overcome these challenges and allow us to probe interactions and structures at a meaningful scale. Let's take for example of a T cell, where its T cell receptors are labeled with a cyan dye. With wide field microscopy that's diffraction limited, we can see that the T cell receptors are labeled, but that's really all we can see. However, with super resolution, we can then begin to resolve individual proteins. And we can see how these proteins are organized at the T cell immune synapse. Protein clusters can be seen, which is very important for T cell receptor signaling, as well as for T cell activation during the immune response. So not only are the individual proteins resolvable, but they are also countable. And their spatial distribution can also be analyzed. Other important ways to use super resolution microscopy include analyzing the molecular composition of subcellular structures. Take here, for example, ion channels. If we zoom in on these ion channels with super resolution microscopy, we can start to see the composition of these ion channels. Some of them are labeled with one type of protein, and some of them are labeled with two types of proteins. We can also understand a bit better the subcellular structures that are present in cells. For example, we can look at nuclear pores, which are important gatekeepers to the nucleus and control gene expression. And they're very tightly controlled because they have a diameter of 120 nanometers with a periodicity of 40 nanometers for each of these nuclear pore complexes that build up this ring. We can also use super resolution to study viral pathogenesis or other small particles. Here we have an Ebola-infected cell, and we can look at these viruses during their assembly as well as during the transport and how they hijack the cellular machinery for their infection. But the challenge is, how can we get more resolution? If we take, for example, extracellular vesicles, these are very small particles. They're about 30 to 100 nanometers in diameter. If we use diffraction limited imaging and label the extracellular vesicles with common markers, we can see that there's a heterogeneous population of the extracellular vesicles. But we can't tell much more than that, even if we zoom in on these images. And using conventional microscopy, like wide field or confocal, we're limited in our resolution by Abbe's law of diffraction, which states that if two fluorophores are less than 250 nanometers apart from one another, then we can't resolve them. Taking, for example, this ring complex of proteins that are very closely spaced together. Because they're so closely spaced together, we activate its fluorescence. What we see is one round spot of fluorescence, but we can't tell how many proteins are there or what structures are there. The solution is to use a super resolution technique called single molecule localization microscopy, where we can activate each and every one of these fluorophores individually and then combine the images to form our structure of this ring complex. And this will give us resolution down to less than 20 nanometers. If we look at this technique a little bit more closely, take, for example, this GFP protein, which is 2.5 nanometers in diameter. If we activate fluorescence, we would expect to see a very small spot. But in reality, what we see is a larger, fuzzier spot called a point spread function, and it's 250 nanometers in size. And we have lots of fluorescent proteins that are located close to one another, and we just see a big blurry spot. What we really want to do is to separate these components into their individual molecules. Now, to do this, we use single localization microscopy, and so we activate each of the fluorophores individually. When we activate those fluorophores, we get this point spread function, which we can then plot on a Gaussian curve. If we find the peak of the Gaussian, we can then localize that to the center of that fluorescent molecule and take down the XY coordinates of that. One single molecule localization microscopy technique is called de-storm, where we use specific commercially available dyes that have intrinsic properties where if you excite them with very high laser power, combined with a special buffer, they enter into a state called the dark state. Fluorophores in the dark state will randomly blink on and off, where at any given time, only a fraction of the fluorophores will be on. And every time they blink, we will take a picture. And then we'll mark each of these point spread functions on a Gaussian and fit them on the Gaussian to get their XY coordinates. If we take many pictures, eventually we can build up the image of the subcellular structure. Here we can see the actin cytoskeleton with a resolution less than 20 nanometers. Now we go back to our extracellular vesicle example. We can ask, what can we do with 20 nanometer resolution? With diffraction limited microscopy, the resolution is greater than 250 nanometers. And if we use TIRF imaging, we can see that there are extracellular vesicles there, but we can't characterize them very well. However, if we perform Gaussian fitting, as I mentioned earlier, with single molecule localization microscopy, then we can detect these single fluorophores and then create a picture that is less than 20 nanometers in resolution. And here now we can start to zoom in and understand a bit better the composition of these extracellular vesicles, their structures, their protein compositions, and how they're organized. And this will give us better insight on how they function in cancer progression and in therapeutics. Conventional super resolution microscopes are very complex. They're expensive and they're hard to use. They require a lot of extra equipment, like a dedicated dark room or an anti-vibration air table. They are very clunky. There's a big learning curve to them and could take a long time to align and get your final image. However, the next generation single molecule platform, the Nanoimager, is Benchtop compatible. You can put it on a Benchtop, as you can see our collaborators here in Hamburg doing, and they put it right next to their biosafety cabinet. We have multiple people run it in a very bright lit room. And so we have one system, the Nanoimager, but it's capable of doing multiple imaging modalities. As we talked about, de-storm is one of the single molecule localization techniques. PALM is a different one that uses photoactivated fluorophores. And these two techniques allow us to resolve and visualize structures down to 20 nanometers. We can also perform live cell imaging and single particle tracking using the built-in tools to explore single particle dynamics and behaviors. We can also perform single molecule FRET, which allows us to study protein-protein interactions. The Nanoimager is equipped with 3 different modes of illumination. On one end, we have epifluorescence, which allows you to see everything in your field of view. On the other end of the spectrum, we have TIRF imaging, which allows you to image close to the cover slip within a 200 nanometer distance. And this gives you the best signal to noise ratio. And then in between, we have high-low, where we can change the angle of illumination to allow us to exceed above that 200 nanometer limit with turf, but still get great signal-to-noise ratio. Many of the core applications that the Nanoimager has been used for by our commercial as well as academic partners include studying extracellular vesicles in cancer progression and therapeutics. They've used the Nanoimager to understand how neural networks can communicate and work with one another, as well as to study pathogens and their pathogenesis during infection. And finally, the Nanoimager has been important for making discoveries in immuno-oncology and CAR T cells. So using the Nanoimager, applications can provide us resolution and sensitivity across multiple orders of magnitude, from single cells all the way to single molecules that make up the subcellular structures within cells. Let's take, for example, cortical neuron networks. We can gain insight on the organization of these cellular networks by performing an overview scan of our networks. And then we can very easily zone in on certain regions of interest to understand how these physical interactions occur within these neuronal networks. And this will give us better insight on how this organization and the geometrical properties and the biophysical properties can work together in the brain for computational purposes. But this doesn't give us very much information on the structural details of individual neurons. So let's dive a little bit deeper using super resolution microscopy. Axons of neurons are very unique in their structure in that they have actin cables that are linked by these rings of spectrin, and the periodicity of these spectrin rings is very conserved, and this is important for their structural integrity. And previously, we weren't able to observe these rings using diffraction-limited microscopy. But now, with single molecule localization microscopy, the Nanoimager is able to resolve these rings very clearly, and we can see that the periodicity between each of these rings is exactly the same as electron microscopy of 200 nanometers. And this gives us better understanding of how the organization of these spectrum rings provides structural integrity to neurons. Additionally, we can dive a little bit deeper to look at individual proteins and link their organization to function. And so here we have neuronal synapses where with diffraction limited microscopy, we see blurry spots. But if we use DStorm, now we can start to understand the composition of the proteins that are at the neuronal synapses, how they're organized, their structural identities, and even how many proteins are at the neuronal synapse. And this gives us better understanding of how the synaptic architecture translates to appropriate signal transmission. We can extend our research beyond cells and look at the research using very small particles using single molecule localization microscopy. If we go back to our example of extracellular vesicles, these are carriers of active biomolecules that have the potential to trigger important biological responses during immune responses, for example, and also during cancer progression. Now, the challenge to characterizing EVs include the fact that the EVs are very small, they're heterogeneous in population, they have variable biomarker expression. So using single molecule localization microscopy, we can then begin to probe their functional pathways, everything from their biogenesis to the type of cargo that they have, to how they can impact and affect other types of cells in therapeutic circumstances, for instance. Using single molecule localization microscopy with our Nanoimager combined with our analysis tools, we can detect unique EV biomarkers. Here we have placenta EVs from a wild-type mouse, and we can tell using single molecule localization microscopy how proteins on these EVs are organized. We can look at the species of EVs and tell which of the population of EVs have which biomarkers. We can count how many molecules of each biomarker is on these individual EVs. And importantly, we can look at the diversity of the population as a whole, but also examine the individual EVs for their unique features. And this method is very sensitive because we can clearly see from knockout models that the signature and protein expression is quite different compared to our wild-type models and negative controls, giving us a way to use single molecule localization microscopy to study the unique features of very small particles. In addition to doing fixed cell imaging, the Nanoimager is also optimized for live cell imaging and single particle tracking. And this is because it comes equipped with temperature control up to 42 degrees. has very powerful single molecule detection and integrated tracking tools that very easily allow you to go from acquisition to analysis right away. It can perform very fast dual color imaging with a high temporal resolution in the millisecond range. So one example of one of our collaborators using single particle tracking with the Nanoimager is by looking at an ER protein called PDZD8 in red here, and looking at how it interacts with mitochondria, because its function is to link interactions between the ER and mitochondria. And we can track the movement of PDZD8 within the cell and build up a map of its tracks, as well as diffusion coefficients. And what's important to note here is that the ER is not labeled. However, we can see the ER architecture just from assembling the tracks of this ER protein. Moreover, we can dive a little bit deeper to look at individual by PDZD8 proteins and its interaction with mitochondria and understand its interaction with organelles. So here we see one of these PDZD8 proteins entering into this mitochondrial cleft, and just by using the single particle tracking system that's in our Nanoimager software, we can start to look at how it interacts with the mitochondria and also see that its diffusion coefficient decreases as it interacts with the mitochondria. And so this was done with simultaneous dual color imaging of both PDZD8 protein as well as mitochondrial marker using live cell imaging, and the tracks were performed using our integrated tracking tools to quantify the particle dynamics, which allowed us to assess the organelle interactions. We can also better understand intracellular transfer pathways of small particles. Going back to our EV example, here we can look at the uptake of EVs by human umbilical vein endothelial cells. And so we can detect cellular uptake of the EVs as well as their internal tracking pathways and map the localization of, for example, encapsulated therapeutics or cargo, and measure their diffusion coefficients of internalized vesicles at different subcellular compartments to better understand how they're being taken up and where they're being targeted to in these cells. Additionally, we can use single particle tracking to understand better the pathogenesis of viruses. And here we can measure the real-time assembly of viruses in a single cell by imaging the top or the bottom of the cell. And so at the apical membrane, if we look at the tracks of the viruses, we can see that they're slower moving compared to if we image them at the basal membrane. And this is really cool because this reflects the state at which the virus particles are in. And they're slower because they're being assembled in the apical membrane, which is a reflection of their slowed down diffusion coefficient. This brings us to an important point about the Nanoimager in studying experiments and in studying virus particles that can be riskier for the experimenter. And so this unique design of the Nanoimager allows experimentation within biosafety cabinets. It has a small footprint. It's a sealed system with remote access with a computer. You can perform live cell research in a BSL-3 or 4 environment, and there's no need to work with virus-like particles because you can evaluate the efficacy of antivirals on wild-type viruses and not virus-like particles. And because it has built-in anti-vibration, it does not need special equipment. You can put it directly into the biosafety cabinet. So one of our collaborators have used this in understanding Ebola virus pathogenesis. And here we see a cell that's stained for actin and cyan, and they've been looking at the virus particle assembly and looking at how it can hijack the actin machinery in order to help it assemble virus particles. But also we can see these virus particles along these actin cables. And this is important for their transport through the cell and eventual dissemination. In summary, imaging with a Nanoimager is made easy because it has multiple illumination modes, high-low, turf, and epifluorescence. It's equipped with 4 powerful laser lines and can perform simultaneous 2-color imaging or sequential imaging if you want to image more than 2 colors. It's benchtop compatible and doesn't require any special room or optical tables. It is a super resolution microscope that's capable of multiple modes of super resolution single molecule localization microscopy. It has a very sensitive CMOS camera for the detection of single molecules. It can perform live cell imaging and temperature control up to 42 degrees. It has a built-in autofocus function for temporal imaging, and it's easy to use with our easy to operate user interface, as well as a digital mirror device that allows you to perform digital confocal. Additionally, we provide a cloud-based analysis platform called CODI, And it stands for collaborative discovery because the goal of CODI is to encourage collaboration across the globe between researchers so that they can easily share data sets across teams and work together to make discoveries. And this tool is equipped with a clustering algorithm that allows you to analyze the protein assemblies in organizations in different subcellular structures. Here we have an example of mitochondria and another example of nuclear pore proteins. You can use this tool even to count the number of molecules that are present in your region of interest. And you can apply filters so that you focus in on the biological phenomenon that you're interested in and apply drift correction algorithms as well. And finally, this tool allows you to build analysis workflows so that you can apply the same parameters across multiple images in batch processing. So the Nanoimager and us at ONI, we provide simple and reliable workflows. We have expert support for sample preparation, experimental design. We provide a desktop-compatible Nanoimager that does not require a vibration table. We provide real-time rendering analysis tools for inspection of the data. And we provide CODI for online analysis and for sharing our data across the globe. So I hope today that I've demonstrated to you that greater resolution provides more information, which ultimately leads to better understanding of biological processes. So join the Nanoimager revolution along with our academic and commercial partners, and encourage you to visit www.oni.bio/applications, where you can then look at protocols, application notes, as well as additional technical resources for using the Nanoimager. So thank you very much. I will pass this time back to Peter.
Thank you, John. Great presentation and tons of questions via the chat panel. So yes, so before we go to Lara, give me 1 second to answer some of them. Yes, 4 lasers are included, 405, 488, 561, 647. The Nanoimager records 2 channels simultaneously, but you can do 4 channels sequential. And if you want to do 3D, we do the astigmatism lens. And Lara now is going to show you how this all works in reality. Over to you, Lara.
Thank you, Peter, and thank you everyone for joining here the webinar on explaining how are the capabilities of the Nanoimager. I will show some of the highlights, and let me start with the first highlight, which is actually that the Nanoimager, the physical Nanoimager, is on the headquarters in Oxford together with the sample, while I am based in Germany and I'm accessing remotely. I will access remotely to the software so that I can perform the acquisition directly from home. So you can see here the feature. This is basically the Nanoimager technical part. So regular table on the lab bench in the wet lab, and lights are on, even the window is open. And you can see here the features. You can start with the Nanoimager. And my colleague Siobhan in Oxford will show us the Nanoimager. This is actually the size of the Nanoimager. She will open now, and you will see that here is the sample that I'm going to image in a minute. That it doesn't have any button, so I will access remotely everything, and I will do everything via the laptop that is next to it. So this is connected with the specific cables, but not like USB cables. And on the left side, you can see the light engine, which is the size of a computer tower. And that's basically it. And that's what you can do. With that, you can do super resolution. So now let's go to the software of the Nanoimager. So this is the software. We have here the visualization window, and here are the controls. My colleague Siobhan has already put a nice sample in it. It has COS7 cells with alpha tubulin stained with Alexa 647. So I'm turning on the 640 laser, and just everything is set for storm. So I will click acquire and just burst the laser. And you can see now this blinking. This is changing because Siobhan has put the imaging buffer, the storm buffer. And when we put the 640 laser at the maximum power, we have this blinking so that we reach now the single molecule localizations like my colleague Ja has explained. So if we go now to the Analyze tab and we look at here, you will be able to see that it's been reconstructed live. So we are having already a super resolution image while acquiring. So the buttons, the controls of the Nanoimager are all here. We have, as my colleague Peter has explained, 4 lasers. So we have up to 4 color imaging, but 2 of them can be done simultaneously. So we have actually the chip divided in 2. And let me check here. If we would like to do dual color, we would turn on this part of the chip. But in this case, we're only doing 1 single color. So it's basically the same chip, but 2 optical paths. So the same field of view, but 2 colors. We also have here the stage controls with the X, Y, and Z. And not only that, we also have here the focusing system, which is also a nice feature of the Nanoimager. So if we go to the left here, we have the focus camera view. And unfortunately, this system does not have a very well centered spot, but this is actually a spot. And this is an infrared laser that focuses at the cover slip next to the sample. So it tells us by focusing this spot where the sample is. Therefore, we do not need any eyepiece, or we do not need to, by a naked eye, to look at our sample. We just focus this spot and it tells us where our sample is. And this is very convenient. Oops, we lost here the window. Let me just go back quickly. So this basically, it's important because if you have transparent samples or things that can be complicated to be focused, this is just a nice system. Moreover, this also allows us to lock the system, the Z-plane, and we just click here, Z-lock, and then it will stay steady for the whole acquisition. We also have the possibility to change our illumination angle. We should, at the beginning, it was at epifluorescence, so the laser going straight to the sample, so you could reach deeper planes on your sample. But in this case, we're putting it at very high inclined illumination or even turf illumination. This means that we will have less excitation in our sample, but this will improve, as you can see, improves very much the signal to noise ratio, which is very important for single molecule localization microscopy. We also have the possibility to do a 3D storm. So here we have an astigmatic lens that we just put at the optical path, and this distorts the PSFs in a way that we can have later on with the algorithm, we can have access to more planes of our sample. And again, like my colleague just said, we have the temperature control here. It can go up to 42 degrees. And if we want to do live cell imaging, we just turn this on, and the whole body of the Nanoimager will be warmed up. This is just the exposure time, and we put it at 30 milliseconds, and this is the number of frames that we are collecting. If you see here, we're reaching almost the 10,000, but I think we are good enough here. So if I just stop here and I turn off the laser, we can go now to the Analysis tab, and you will be able to see here the difference between wide field microscopy and the reconstructed the storm image. So this is clearly the difference between the 2 modalities of imaging, and we win by 10-fold our resolution. This software also has some primary analysis capabilities. Like, for example, I will show you here a feature, which is a line histogram. So let me just put here an area that I will just collect. And if I enter here, you can clearly see the separation of the microtubules. Whereas if we look at the wide field, it is really hard to follow. So we have here the 3 microtubules. We can extract here the information of, for example, the distance between the microtubules, or the size, the width of the microtubules. And as you can see, if you imagine that the wide field microscopy is about 200 to 150 nanometers in resolution, and we are talking about here less than 200 nanometers, and this is the power of the storm in this case. And this can be done with the Nanoimager directly. We also have other capabilities on this software for example, light cell imaging, single particle tracking. Imagine that if we had a particle that would be moving and you would like to follow this track, in this case it's not. We would have here the parameters to run the tracking. Some tracks will appear here, and then you can extract information such as the speed, so the diffusion coefficient, the track length, how long they are, these tracks, the size of the particle, or even the concentration of our sample. All this is possible with the Nanoimager, but I will not have a sample here today. And the same goes for single molecule FRET. We have here a software that allows us to do analysis on the single molecule FRET imaging with all the parameters that you need to extract. Moreover, we still have some more features, and this is actually the overview scan. So this is, you have to imagine that we have at the moment objective that is 100x in magnification. So how can we see if our sample is actually looking nice or not? We can have this possibility of the overview scan. You can see already here that my colleague has done some, but I will run quickly a new overview in which you can tile different fields of view and just look at your sample as if it was a lower magnification objective. So now we can see here, where are we, whether we would like to go to different positions. I can go to this position and then save it, and then go to another position, and the Nanoimager reacts immediately. Anything that is interesting for us, and then we can save that for a later acquisition. More than that, if you would like to do a more elaborated acquisition, such as, for example, like as I said, multiple positions or Z-stacks, this is also possible. We go to this multiple acquisition setup, in which you can set here different acquisitions, such as if you would like to do multiple setup, like different positions, or again, if you would like to acquire the Z-stacks or perform a time lapse, all this is possible. You would just program everything here, click on start, grab a coffee, and come a few minutes later, and all the acquisition will be done for you. And this is pretty much everything that we can do with the Nanoimager. Let me now go to the cloud-based platform that we also have, in which you can perform more deeper analysis. So let's see. So you have to imagine this is not only capable for microtubules, we can do other things here. And this software allows you to do many things, such as clustering or other things that I will just explain in a minute. This is just a tubulin image, the same that was acquired a few minutes ago, but just some time ago, some days ago. And this is the platform. So this is cloud-based. I'm accessing at the moment in the browser, and this is what it would look like. This data set, I would upload it and have it here with different parameters here that tell me the information. But not only that, I can also share it. I say this at the moment, this data set is private, but I could just look at my colleague Ja. She has access to Cody, so I would give her access as, I have to go back. Sorry about that. So I would just share it with my colleague. Just a second. And then I could give her access as a viewer, as an analyst, or just share ownership. Or if I think that actually this data set is really nice and I would like to make it public for everyone that has access to Cody, I could also make it public. But not only that, as I said, this is capable of doing some clustering. And I will just go to another data set that we have here. And this is actually extracellular vesicles that my colleague has explained. This would be one extracellular vesicle. And here you can see the different possibilities that you can have for analysis, starting with the drift correction. So if this is drift corrected, if I would just remove it, you would see that the image is not, it's a little bit drifted, but I can put it back on. And then it very nicely reconstructs the EV in a decent structure of the three colors. So let me just say, here we're doing three-color storm, in which we have three tetraspannings for these EVs, CD63, CD81, CD9. And then we will need some parameters, because this was done sequentially, so that the first color was actually the Alexis 647, which is the CD81, then the CD63, and then the CD9. So I don't want to collect possible localizations that crosstalk. But the good feature of that is that since I can share it, I can actually load a filter result that was done from my colleague Andras at ONI. I just upload the filters, and I don't need to spend any time just optimizing the filters. Somebody else has done it for me, and now I have the possibility to continue with my clustering. You can also set an ROI if you would like to set, like, for example, a specific area, and only this area would be analyzed. You can also do squares. You can also do polygons. This is also possible. But the really nice feature here is the clustering tool, in which, again, I could run it, but for the sake of time, it's actually
your computer screen.
Sorry?
Please put on your computer, yeah.
So now I am just accessing to the clustering result that my colleague Andras has done some time ago, and he has already set specific parameters. And you can see here the cluster. So this is just given here. I can just turn it on and off, and this is very nice. And now if I click on the cluster, I can get information on the localizations and on the area of these AVs. And again, if I want to optimize my parameters, I could play around with the different parameters that we have, with such as area localization. An important one is the circularity of the EVs. So we want only circular EVs. We could move that. But again, my colleague Andras did already this optimization. And now you will be able to see that he has moved the area that makes more sense for EVs, removing super tiny EVs that might not be EVs, but just debris, or super big ones that might be just aggregates for the skewness. He has also set the specific points. And again, we can extract this information. Moreover, you can also think, okay, and can I get counting? I have seen some of the questions were about counting. Well, we can do counting of the localizations. These will not be absolute numbers of the proteins in the EVs, because we are here having labeling with antibodies, and antibodies can have multiple. They can be polyclonal, they can be monoclonal, and they can have actually 1, 2, or 3 fluorophores, but this will depend on the labeling strategy. But what we can do is actually extract the stoichiometry and the positivity, for example. So this is actually the counting tool. Again, I will load the counting tool that my colleague Andras did some time ago, and I will use this to explain you what I mean by that. So now, again, we still have the clusters here, but if I click now, it tells me another information, which is the number of localizations for each of the tetraspannings. This empty channel, just ignore it. You don't have to think about it. I will not explain here, but it has to do with the channels on the chip. I said the dual color. This is just this thing, but forget about it. So again, if you look at the tetraspannings here, we can measure them, and here gives you a possibility to look at the different, for example, the different EVs that contain CD81 and CD63. So if I click here to another EV, it will tell me, okay, it has the triple color. This is another EV. This is another point on this scatter plot. All this information can be extracted. And you have to think about it, that it is in fact not only specific for EVs. If you are imaging membrane receptors and you would like to look at colocalization of these membrane receptors, this can be also a very interesting tool for that. So with all that, I will leave it here and I'll be happy to answer any more questions. But first, Peter, I'll give you back the word. Thank you everyone for assisting to this live webinar.
Thank you, Lara. Great demonstration. I think the audience is really excited. You got many, many compliments. Also, Ja, you also got many compliments. Well done. Well done. So before we start to the questions and answers, I do a commercial break to give you some rest. So our products are the ONI Nanoimager. We also have the EV workflow kit. We have a B-Cube buffer. And if you want to have a demonstration of the Nanoimager in your lab, go to the website and push the Get in Touch button. Then the form comes and then you can request a demonstration. Okay, so now it's question time. Before we start answering questions, I will say that there are so many questions, it's impossible to answer them. We are running also a little bit late. We will make a Q&A summary of all the questions and answer them, and we'll send them around to all the attendees. Okay, I hope you're happy with that. Okay. So there was a question about how many fluorophores binding to view a 20 nanometer EV. Maybe Lara, this is a question for you to answer.
Yeah, so as I said, it depends on the binding strategy. So in this case, if it's usually just antibodies, it can be primary antibodies already conjugated with the fluorophore. This would be less. It can be antibodies like primary with secondary that are labeled with the fluorophore. This can give you more points. So what we can extract actually is the stoichiometry, as I have just shown. For more absolute numbers, we will have to think on a more specific strategy for extract this information.
So the next question was, are there any special requirements in terms of fixation, staining protocols? Maybe this one is for Jan?
Yeah, so there are lots of different staining and fixation techniques. It really depends on the subcellular structures that you're interested in looking at. But these can be very common for not only for super resolution imaging, but also for conventional microscopy. And so our website, applications portion of the website has a lot of different tools and resources and protocols for protocols that allow you to use different kinds of fixation to see the structures that you want to look at. And we're also available as field application scientists and our customer success team members. We're available to help you with your experimental design.
Thank you, John. And then there is some question about software. All the software you have seen, 2D and 3D, is all developed by ONI staff. So it's ONI proprietary software, but it's available for everybody. All the system works in 2D and 3D, and also on the website, as Jar was explaining, there's lots of information, and also there's a list of all compatible fluorophores. Next question is about binding, principle of binding. Maybe Lara, this one is for you.
Yeah, so usually you have a specific antibody. So if you are talking about immunostaining, you have specific antibodies that are against a specific protein, and this it will bind to the protein of interest, right? And then it can depend. You can have directly a conjugated fluorophore next to the antibody, and then you would just put it for, let's say, 1 hour on your sample, and then these antibodies would bind. You can also have a primary and secondary antibody so that it's more specific. Like the primary would be against the protein of interest, and then the secondary would be against the primary antibody, and then you would have, again, the fluorophore. This is how DStorm works. There are other capabilities also for single molecule localization microscopy, such as transient binding. This is paint in which just basically a specific protein attached with fluorophore transiently binds to the target, and then this will give you also super resolution information, but also dynamics. And then there is fluorescent proteins, but special fluorescent proteins called photoactivatable or photosoluble fluorescent proteins, in which you can genetically encode the protein of interest that will your cell will be able to express together with this fluorophore. And then this fluorophore will only be will change its spectral state with UV light, so that you can control when this fluorophore will come out. This is very helpful if you want to have information on the single molecule, because if you are doing imaging at the full like regular wide field, you usually have the fluorophores all on at the time. But the feature of the super resolution on the single molecule localization microscopy more specifically is to be able to extract this information, the single molecule, and this is by separating in time the signal. And then with palm imaging, this is also possible.
Okay, there was one question about the size. The Nanoimager is small. It's 21 by 21 by 15 centimeters, but the lasers are not included. Maybe you saw in the live demo that Lara gave about the overview of the system, there was a black box like the size of a computer. And in that box, there are the lasers. Maybe, Lara, you also can answer the question about the size of the EVs. How do you quantify the size of the EVs?
So, yeah. So the software has some of the different parameters that I just briefly showed. Some of them is actually the length or the radius. So maybe I can just go back quickly and show you briefly. Let's see. If I go to the clustering tool and I just look at one EV, if I look at the radius here, oops, this is off, so we'll turn it back on. So you see the radius, it's about 45 nanometers, for example, for this EV. If I go to this other EV, I could see, well, this might not be, this might be just a tiny cluster. So actually, this probably is just the radius of this cluster. So if I remove, in fact, the noise, it is not the size of the full EV, it's just the size of the cluster attached to the EV. So the protein of interest, this is a tetraspanin. In this case, it's probably CD63 that it has cluster inside the EV. And then you can look at this as the cluster, and this would be the size of the radius. So the software, the CODI, can actually extract the information on the size of the EVs.
Thank you, Lara. I think there are some questions about the EV. Yes, we offer the EV kit for that, and the EV kit has cartridges, and unfortunately the cartridge is not reusable. So like I said, we are past the hour, so I think this is a good moment to end. And I would like to thank you all for joining us. You were very active on the questions, so I promise you we make a summary of all the questions, and you get a written answer via email from us. Okay? I have one more request to you. Please keep visiting our website for all the information. We have lots of app notes there. We have sheets. We have applications, how to do. And then please keep following us on Twitter and LinkedIn. We will communicate the next webinars on there. And one question that I got privately in the chat, what are the topics of the next webinars? And of course, based on the questions that you are asking, EV must be a topic of the next webinar. Okay, thank you all for coming and see you next time. Bye-bye. Thank you, John, Jack. Thank you, Lara. Bye-bye.
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 Bye, everyone. Thank you.